Okay, so welcome to this next video in which we are discussing adrenoreceptors. Okay, right. We've just finished our discussion of the alpha-1 adrenoreceptors. What we want to now move on to is discussing the alpha-2 adrenoreceptors. Okay, and we know that there are, again, 3 types of alpha-2 adrenoreceptor, which are all coded for by different genes. Okay, so we have the alpha-2A adrenoreceptor, the alpha-2B adrenoreceptor, and the alpha-2C adrenoreceptor. And we want to see what these adrenoreceptors are going to do, where they're positioned and what they're going to do. So basically, let's start with where they're positioned and what they're going to do, and then we'll discuss how they do it, which heterotrimeric G proteins they are coupled through, and how that takes effect. Okay, so where are they located then? So basically, in the human body, there are neurons which release noradrenaline. Okay, so let's have a neuron which releases noradrenaline here. Okay, so this is some noradrenergic neuron. So this is a noradrenergic or norepinephrine noradrenergic neuron. Okay, so it releases noradrenaline. Now basically, alpha-2 adrenoreceptors are located on the presynaptic neuron itself. Okay, so you might have an alpha-2 adrenoreceptor here on the neuron itself, and the purpose of this alpha-2 adrenoreceptor is negative feedback. Okay, so the presynaptic neuron is going to release noradrenaline, which will act on the next cell, whether it's another neuron or whether it's some other target cell, such as a smooth muscle cell, for instance. But you also have a negative feedback system where the noradrenaline works on receptors that are called autoreceptors. Okay, so auto means self. So for instance, autonomy means self-governing. So autoreceptors mean receptors on yourself. So these are receptors for the neurotransmitter that are present on the cell which has just released the neurotransmitter. So this is an autoreceptor. So basically, these alpha-2 adrenoreceptors are generally autoreceptors. They're on the surface of the cell which releases the noradrenaline. And what do? Well, basically, they inhibit the presynaptic neuron from being able to release any more noradrenaline. So they inhibit the release of adrenaline, basically. Okay, so negative feedback is their purpose. So when the noradrenaline concentration gets to a certain height within the synaptic cleft, it will feed back on the alpha-2 adrenergic receptors, and that will cause a negative feedback of the release of more noradrenaline. Okay, right. So this auto-inhibitory negative feedback is the main it's the main localization of these alpha-2 adrenergic receptors, and this is the main function of these alpha-2 adrenergic receptors. Okay, so what we want to look at in this video is how these alpha-2 adrenergic receptors achieve this auto-inhibition. How do they stop the presynaptic neuron from releasing more neurotransmitter? Okay, so let's start by looking at the heterotrimeric G protein which alpha-2 adrenergic receptors activate. Okay, so let's draw our alpha-2 adrenergic receptor here. Okay, and for this entire discussion, we will not distinguish between the 3 different types of alpha-2 adrenergic receptor. All of them are located presynaptically like this and are involved in this auto-inhibitory negative feedback. So we'll just call them alpha-2 adrenergic receptors. Okay, so here is our G protein coupled receptor. Here is our amino terminus, here is our carboxylic acid terminus, and let's say a noradrenaline molecule has come and bound to the transmembrane region of this alpha-2 adrenergic receptor and has hence activated it, because remember, it is a rhodopsin family G protein coupled receptor, so the ligand for it binds within the transmembrane domain. Okay, and the binding of this noradrenaline, okay, so this is noradrenaline, will trigger a conformational change in the alpha-2 adrenergic receptor, so the alpha-2 adrenoreceptor, and what it will do is it will make available a binding site for a heterotrimeric G protein. Now, which heterotrimeric G proteins do alpha-2 adrenergic receptors bind to? Well, generally it's a GI heterotrimeric G protein, and what this means is that the alpha subunit of the heterotrimeric G protein is generally a G-alpha I subunit. So remember, there were 3 separate G-alpha I subunits. There was G-alpha I1, there was G-alpha I2, and there was G-alpha I3. This doesn't tell you which one of those specifically it is. It tells you that it's one of those 3, basically. And of course, it doesn't tell you at all which beta or gamma subunit we are talking about here, because that sort of information just isn't known, basically. Okay, so here is the beta-gamma subunit, and then we've got some sort of alpha I, whether it's alpha I1, alpha I2, or alpha I3. Okay, so let's colour in the alpha I subunit in red, and at present, the way I've drawn it is the alpha I subunit is turned off, and is therefore bound to the beta-gamma subunit, so I need a GDP molecule bound here. Okay, so let's colour in the beta subunit in blue here. Okay, and then we'll have the gamma subunit in green. Okay, so in green here, this is the gamma subunit. Right, okay, so basically what's going to happen then is this heterotrimeric G protein will be able to bind to the alpha-2 adrenoreceptor once the noradrenaline has bound to the alpha-2 adrenoreceptor. Okay, and it's the alpha I subunit that will actually bind in here. Okay, and alpha-2 adrenoreceptors only bind to heterotrimeric G proteins in which the alpha subunit is of the alpha I form. Okay, so the heterotrimeric G protein will go in and bind, and then what will happen, of course, is the old story. The GDP will come out, the GTP will go in, and then, of course, when the GTP binds, the beta-gamma subunit cleaves off, and then once the beta-gamma subunit's gone, the alpha subunit can no longer bind to the intracellular aspect of the G protein coupled receptor, so it also goes off. So we produce these 2 active subunits then. We produce the beta-gamma complex here, that's still held in the inner leaflet of the phospholipid bilayer by this prenyl group of the gamma subunit, okay? And we also produce this alpha I GTP subunit. And both of these will have downstream effects. Now, usually when we talk about alpha I pathways, we will then go on to talk about the alpha I subunit itself and how it inhibits adenylyl cyclase enzymes and how important that is. In this case, however, it's the beta-gamma complex that is actually more important in presynaptic negative feedback that we're talking about. Okay, so we're actually going to abandon the alpha I subunit. It will go off and inhibit certain adenylyl cyclase enzymes, but we're actually going to focus on the beta-gamma complex because it is the one that actually produces the presynaptic negative feedback. Okay, so we're going to focus on this subunit here, basically, the beta-gamma complex, and we want to see how is it going to stop the presynaptic nerve terminal from releasing more noradrenaline. Okay, right. So, it's going to have 2 main actions that I'm going to tell you about. Okay, it's going to cause hyperpolarization of the electrical potential difference across the cell membrane, okay, of the axon terminal. That will make it more difficult for the action potential to propagate into the axon terminal. It's also going to inhibit the voltage-gated calcium channels. Okay, right. So, what I'm going to start off with is a little discussion of how you actually release neurotransmitter from nerve axon terminals, and then we'll come back to these two things. And because once we understand how neurotransmitter is released from axon terminals, we'll be in a better place to understand how these things are going to block the release of neurotransmitter. Okay, so let's draw another nerve ending here. So here is our axon terminal, and here is our axon entering the axon terminal. Okay, so basically what happens is the action potential will propagate along the axon terminal, okay, and it will propagate along the axon, and it will propagate into the axon terminal. Now let's just discuss the action potential in a little bit more detail. So what actually happens when an action potential occurs across a portion of membrane? Okay, so let's say this is a portion of membrane here, and we want to discuss what actually happens in an action potential. Okay, so usually the electrical potential difference across the cell membrane is around negative 65 millivolts. Now, what does that actually mean? Well, basically, electrical potential is a concept from physics, okay? It is a mathematical model to understand the real world around us, okay? And this is basically the mathematical model that you use. You ascribe to every point in three-dimensional space a real number, okay? So it could be negative, it could be 0, it could be positive, it can be any real number. It doesn't have to be an integer, it doesn't have to be rational even. It can be pi or e or one of these silly numbers, okay? It can be any real number. So you ascribe to every point in real three-dimensional space a real number, and that number is going to be called the electrical potential. And the units of electrical potential are volts, okay? So you ascribe every point in space a certain electrical potential. Okay, now, basically, all the points inside the cell, they will all become in equilibrium with one another, and they will all get the same electrical potential. Okay, so all the points inside the cell will have the same electrical potential. So in principle, I could get you a little man, okay, so let me draw this little man, okay, and he can have a machine that measures electrical potential, okay, and he can go inside the cell, and it does not matter where he stands, because all of the points inside the cell will have the same electrical potential, and he can therefore measure this electrical potential intracellularly. And I will call that the electrical potential, big E, and then I will subscript it I for electrical potential intracellularly. Okay, so this is some one number that he will get no matter where he stands within the cell. Now, the extracellular fluid will also be in electrical equilibrium with all the points in the extracellular fluid will also be in electrical equilibrium. So again, they are all at the same electrical potential. So this little man could go into the extracellular fluid, and he could also measure the electrical potential of the extracellular fluid, which I will denote big E for electrical potential, and then subscript to E for extracellular. Okay, so basically, you can go into the extracellular fluid and there will be a certain electrical potential there, and in the intracellular fluid there will be a certain electrical potential there. Now, these two numbers are not necessarily the same, so the extracellular fluid is not in electrical equilibrium with the intracellular fluid because of the membrane that's in the way, basically. This means that you don't have to have them in electrical equilibrium. Okay, so basically, there is therefore an electrical potential difference, or a voltage, across the membrane. So voltage is just another name for electrical potential difference, a difference in electrical potential. And strictly speaking, what you should do whenever you talk about voltages is you should say the voltage, or the electrical potential difference from one place to the other place. So strictly speaking, what I should say is the electrical potential difference from the extracellular fluid to the intracellular fluid. Okay, this is what people usually mean by the electrical potential difference across the cell membrane, but people get sloppy and forget to tell you what orientation we are talking about. Okay, it's assumed that everyone knows, basically. So, what does this actually mean? This means if the little man was to move from extracellular to intracellular, how much would the number on his machine change? So let me say that again. He starts extracellularly. He gets some number on his machine. Then he somehow gets across the cell membrane and goes into the intracellular compartment, and then gets another number on his machine. What we are now asking is how much has the number changed on his machine from going from extracellular to intracellular. That is what I mean by the electrical potential difference from extracellular to intracellular, or the electrical potential difference across the membrane, or the voltage across the membrane. So what is this number? Well, this is the new electrical potential that he's experiencing, the new number on his screen, which is the intracellular electrical potential. Subtract the old number that used to be on his screen, which was the extracellular electrical potential, which was big E subscript E. Okay, so this is what is meant by the electrical potential difference across the cell membrane. We mean the electrical potential difference when you move from extracellular to intracellular. Okay, so this number is usually negative 65 millivolts. Now, what does that mean? That means that the electrical potential of the intracellular compartment is lower than the electrical potential of the extracellular compartment, and it's lower by 65 millivolts. So this number is 65 millivolts smaller than this number, basically. Okay, right, and that's the normal electrical potential difference across the cell membrane. Now, basically, in an action potential, what happens is some initial stimulus occurs that causes depolarization, okay? And we'll talk about how it propagates in a moment, but initially what's going to occur, and I'm finding this difficult at the moment, do I want to explain it from the beginning, or do I want to explain it how it propagates? Okay, I will explain it from the beginning. Okay, so some initial stimulus occurs that will cause some sort of depolarization of the membrane. So maybe some sodium channel opens, okay? So for instance, many ligand-gated ion channels are sodium channels, okay? So for instance, it could be a glutamate receptor. You can imagine it's a glutamate receptor, okay? And when this glutamate receptor opens, what will happen is it will conduct sodium ions from the extracellular fluid into the intracellular fluid, okay? Because sodium concentration extracellularly is much higher than sodium concentration intracellularly. It's around 145 millimolar extracellularly, whereas in the intracellular fluid it's around 12 millimolar. In addition, sodium ions are a positively charged ion, okay? So they want to be where the electrical potential is lower, and currently the electrical potential is lower in the intracellular fluid rather than the extracellular fluid. So, when you open this ligand-gated ion channel, which could have been opened by glutamate or something, what that will cause is it will cause some sodium to move into the cell, basically, from the extracellular fluid. Now, what will happen when you do this? Well, you're bringing positive charge into the intracellular fluid. Okay, positive charge increases the electrical potential of an area, so the electrical potential intracellularly will go up. Meanwhile, the electrical potential extracellularly is going to go down because you're taking positive charge out of the extracellular fluid and into the intracellular fluid. So when you remove the positive charge, that's going to cause the electrical potential to go down. When you dump it into the intracellular compartment, that's going to cause electrical potential to go up. So this one goes up, this number goes down. What effect is that going to have on the electrical potential difference across the cell membrane? Well, basically, originally, the intracellular electrical potential was lower than the extracellular electrical potential by 65 millivolts. Now, this number is going down, this one is going up. So if we now ask how much lower is the intracellular electrical potential than the extracellular electrical potential, the answer is going to be, it's going to be less, the amount by which it's lower. Okay, so this number is going to become less negative. Okay, and that is called depolarization. So when you make the electrical potential difference less negative, that's called depolarization. So when the amount by which the electrical potential goes down, if you move from extracellular to intracellular, becomes less, that's called depolarization. Okay, so let's draw a little graph of this. So we will plot the electrical potential difference from extracellular to intracellular on the y-axis versus time on the x-axis. So at the initial time, let's say it was at negative 65 millivolts down here, but now we have stimulated the membrane with glutamate or something, and that's causing this depolarization, so the electrical potential difference across the cell membrane is getting less negative. Okay, and when it rises, if it rises, of course, there's a big if there, if it gets to negative 40 millivolts, then it will trigger the membrane to undergo an action potential. Now, what happens in an action potential? Well, basically, what will happen is the voltage-gated sodium channels in the local area will start to open. Okay, so let's say this is a voltage-gated sodium channel in pink here. So this represents a voltage-gated sodium channel. Okay, so what will happen is when you get the electrical potential difference across this membrane to negative 40 millivolts, which means that now the intracellular electrical potential is only 40 millivolts lower than the extracellular electrical potential. This voltage-gated sodium channel here, which I'll just abbreviate to VGNC, for voltage gated, and then the N is for sodium, that's using the periodic table symbol for sodium, and then the C is for channel. This will open, and this will now do the job of the glutamate receptor, or the initial depolarizing source, better, basically. You'll have loads of these, not just one, there'll be many of them in this little portion of membrane, and they'll all open. And they'll all allow sodium ions to move in from the extracellular fluid. Okay? And now, of course, what do sodium ions do? Well, they cause more depolarization. So you're raising the electrical potential of the intracellular compartment even higher because you're bringing in more positive charge, so it's going to go up, up, up. And the electrical potential of the extracellular compartment is going to go down, down, down. Okay? Because we're taking positive charge out of there. Okay, so negative 40 millivolts is the point where these voltage-gated sodium channels become activated to open. And after that, what you'll get is a very steep depolarization of the electrical potential difference. And it will even get to positive levels, which means that now the intracellular electrical potential is higher than the extracellular electrical potential. And it generally gets up to a level around plus 40 millivolts, basically. Okay, right. Now, at that point, the voltage-gated sodium channels will close. And I want to stress that it is not the voltage that closes them. They don't close because we've got to plus 40 millivolts. They close just on a time basis. So they think, we've been open now for long enough, we'll start to close. Okay, so that stops any further depolarization now, because you're no longer letting sodium into the cell. Okay, now what you're going to do is start letting potassium ions out of the cell. Okay, now this is because the voltage-gated potassium channels, certain voltage-gated potassium channels, are going to start opening. So here, let's say this can represent a voltage-gated potassium channel here. Okay, we'll colour it in orange. So this is a voltage-gated potassium channel, and basically the voltage-gated potassium channels are generally opened by the same threshold potential as open the voltage-gated sodium channels. So this is an important concept. They were activated right down here. okay, at the same threshold potential or thereabouts as the threshold potential which opened the voltage-gated sodium channels. The difference between the voltage-gated potassium channels and the voltage-gated sodium channels is that the voltage-gated sodium channels, as soon as the electrical potential difference across the membrane got to negative 40 millivolts, they were like, fantastic, get ourselves open instantly and let's depolarize this membrane. Whereas the voltage-gated potassium channels, they were kind of a bit more pathetic. They said, oh good, and then sort of sat around for a bit and then said, oh, we'll finally open. And by the time that they actually get around to opening, the voltage-gated sodium channels have done their job and they've closed now. Okay, so at about this point, the voltage-gated potassium channels actually get around to opening. And I shouldn't have drawn that yet. I haven't actually explained what's going to happen yet. So, what happens now? Well, the concentration of potassium intracellularly is around 155 millimolar. The concentration of potassium extracellularly is around 4 millimolar. Okay, so potassium's concentration gradient says, let's move potassium out now, we've got this channel open. Okay, however, we also need to take into account the electrical potential difference across the membrane. At the moment, we're at positive levels, okay? So, that means that the electrical potential intracellularly is actually at the moment higher than the electrical potential extracellularly, okay? So, potassium ions are a positively charged ion. They want to be where the electrical potential is lower, which is currently the extracellular fluid. Okay, so not only is the concentration gradient favouring the movement of potassium out, but also the electrical gradient is favouring the movement of potassium out. So what will happen when you open these voltage-gated potassium channels is that potassium ions will move back out. Well, they'll move out of the cell, basically. Okay, and that means that you're going to reduce the electrical potential intracellularly because you're removing positive charge from the intracellular compartment, and you're going to raise the electrical potential extracellularly because you're putting in positive charge into the extracellular compartment. Okay, and this is going to mean that the electrical potential difference repolarizes. Okay, so you're going to go back to more negative levels. Okay, so back to more normal levels. Right, and even once we actually get the electrical potential back onto the negative levels, you're still going to get the movement of potassium out. The reason being that the concentration gradient will overwhelm the electrical gradient. So for instance, if we're at, let's say, negative 20 millivolts, which is about what this looks like, that will mean that the potassium ions' electrical gradient is telling them to stay in the intracellular compartment because now the electrical potential intracellularly is 20 millivolts lower than the electrical potential extracellularly. However, look at the concentration gradient. The concentration gradient overwhelms the electrical gradient at that level. To get an electrical gradient that will be strong enough to completely oppose the concentration gradient driving force, you would have to have an electrical potential difference across this membrane of around negative 85 millivolts, the Nernst potential for potassium. Okay, so at negative 20, it's nowhere near strong enough to completely stop the movement of potassiums out, and we're never ever going to get down to negative 85, so we don't need to worry about that. Okay, so what will happen is these voltage-gated potassium channels, which are open at the moment, will repolarize the membrane nicely. You'll continue to get potassium ions moving in, and then they will give up. They, like the sodium channels, will get fed up after a certain amount of time, and they will close. Okay, and then what happens is you're just back to the normal leaky channels being open. So you're back to the normal sodium channels and the normal potassium channels, and what will eventually happen is you'll re-establish an equilibrium, basically, and that's the action potential. Okay, now, how does this propagate from one piece of membrane to the next piece of membrane? Well, basically, if you've got an action potential occurring, let's say, in this orange piece of membrane of the axon that is about to propagate into the axon terminal, what will happen is when this membrane is in the depolarizing portion of the action potential, and you've got sodium ions coming in through these voltage-gated sodium channels, some of these sodium ions will diffuse over to the neighboring portion of the cell membrane, okay, and they will go into the intracellular fluid here. Now, what will they trigger? They will trigger depolarization of that membrane. Okay, why? Because you're bringing in positive charge into the intracellular fluid. So, the electrical potential intracellularly will go up. Okay, so that number will go up. Okay, which means that when we asked what's the difference from extracellular to intracellular, the number is going to get less negative because this number is going up. If you want that in terms of the equation, if this number goes up, the answer to this is going to become less negative. And of course, we're leaving this one roughly the same. So, what that will trigger then is the depolarization of the membrane, and if that gets up to negative 40 millivolts, threshold potential for the activation of the voltage-gated sodium channels, then this portion of the membrane will undergo an action potential. Okay, so then you've got this portion of membrane undergoing an action potential, and then the idea is it can propagate from this portion of membrane to the next one in the same way. Now, what we're going to see is that the beta-gamma subunit is going to cause the hyperpolarization of the membrane of the axon terminal. Now, what does that mean? That means that the normal resting electrical potential difference across the cell membrane is going to be more negative than negative 65 millivolts. Okay, so we might take it down to something like negative 75 millivolts, let's say. Okay, and if the electrical potential difference across the cell membrane is taken down to negative 75 millivolts, do you think that this portion of membrane will find it easy to spread the action potential to this portion of membrane? Well, the answer is no, because you're going to give it the same amount of sodium, so the same amount of sodium will diffuse over. But now, if we're at negative 75 millivolts initially, so down here now, okay, we're going to have to produce a bigger depolarization of the electrical potential difference across the cell membrane to get it up to negative 40 millivolts so that we can then fire an action potential. So it means that the membrane becomes more difficult to stimulate. You're going to have to give it more sodium, and if there isn't enough sodium to actually cause that depolarization up to threshold potential, then it doesn't matter. You're not going to get the action potential spreading into the axon terminal, and then the axon terminal won't release neurotransmitter. So even if action potentials are continuously shooting down this axon, if they can't get into the axon terminal will not release neurotransmitter. So you effectively shut the axon terminal down and say, we don't care, axon. You can fire as many action potentials as you like, but we're not going to release neurotransmitter. Okay, so that's one of the effects of the beta-gamma subunit. In addition, if we think about the other portion, if we think about how having an action potential propagating through the axon terminal actually leads to the release of neurotransmitter. Okay, so we have docked synaptic vesicles at the presynaptic membrane here. So these synaptic vesicles here are full of neurotransmitter. Okay, then we know that basically the way that an action potential causes the release of these synaptic vesicles is basically that you have special channels, which we're going to study in a lot of detail in a moment, known as voltage-gated calcium channels, VGCCs for short. Okay. Basically, these will be activated when the membrane undergoes depolarization. So if this membrane fires an action potential which contains the voltage-gated calcium channel, then at some point the membrane potential will get, the electrical potential difference across the membrane will become highly depolarized. This will activate these voltage-gated calcium channels, which will then open and allow calcium to move from the extracellular fluid, in which, remember, the concentration is around 1.5 millimolar, into the cytoplasm where the concentration is closer to 100 nanomolar. and this calcium will then be sprayed onto the machinery of the machinery that fuses the synaptic vesicles with the presynaptic membrane. And the synaptic vesicles will then be fused with the presynaptic membrane, and you'll get the exocytosis of the neurotransmitter. The beta-gamma subunits are also going to bind to these voltage-gated calcium channels and cause inhibition of them, i.e. even if you do get the action potential getting into the axon terminal, then you won't actually be able to activate the voltage-gated calcium channels, you won't get the calcium coming in, and therefore you won't get the release of the synaptic vesicles. Okay, so by these two mechanisms, the beta-gamma subunit being active in these synaptic terminals is going to stop the release of the synaptic vesicles. Okay, right, so we'll call it there for this video, and in the next video we will look in detail at how we produce hyperpolarization, and we'll also look in detail at the structure of the voltage-gated calcium channels.